to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Liquid Class Optimization, Tips and Tricks for Efficiency and Reproducibility. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank Nova Biomedical, who developed the content and brought us together today. Advanced Instruments and Nova Biomedical are now doing business under the same brand, bringing together decades of expertise in analytical instrumentation, R&D, and global customer support. The Artel product line is now part of Nova Biomedical's portfolio, providing liquid handling volume verification products and services to life science laboratories worldwide. Now I'd like to introduce our speakers for today's event. Throughout his 20-year career at Hamilton Company, Eric Sindelar has been instrumental in the adoption and implementation of liquid handling automation technologies. In his current role as Director of Applications, he oversees applications efforts in North America with a team of over 80 field specialists, automation engineers, and trainers. As applications team lead, Samantha Turnbull supports a team of field application specialists in advancing laboratory automation and liquid handling solutions for clients across the life sciences sector. She has a background in immunology research with a master's degree in biomedical engineering. And Nick Ania is the product manager for the Artel product portfolio under Nova Biomedical. With more than 10 years of experience as a laboratory application scientist with Artel, Nick brings deep expertise in genomics, immunoassay development, and regulatory compliance. Having worked in the molecular diagnostics and instrumentation space, Nick understands the issues involved with taking a regulated product from research to the market. Now, before we continue and we hear from our first speakers, we'd first love to hear from you, our audience, with a polling question. This should be appearing on everyone's screen right now, and if you're attending via GoToWebinar, you can participate by selecting on any of the answers you see in front of you and then clicking Submit. The question is, which type of Hamilton liquid handler do you currently use in your lab? Your answer options are Microlab Prep, Microlab Nimbus, Microlab Starline, Microlab Vintage, or other. And we'd love to hear your thoughts on the other in the questions window, and we take in your thoughts there. Now, again, the question is, which type of Hamilton liquid handler do you currently use in your lab? I'm going to give everyone a few more seconds to consider an answer and participate, and in a moment then after that, I'll hand it to our first speakers. All right, it looks like we have most of our answers in. Thank you everyone for participating in this. Let's go ahead and look at the results for this. We have 55% of you selecting for Microlab Starline, then 18% for other, 14% for Microlab Vantage, 9% for Nimbus, and then 5% for Prep. So again, thank you so much for your participation. But now, without further ado, I'll hand things over to our first speaker, Eric Sindelar. You may begin when you're ready.
Great, thanks so much, Ryan. Really appreciate the answers to the poll questions. Great to see so many Hamilton users attending the webinar. For those that may not be using Hamilton platforms or may not have adopted any automation technology, know that the topics and functions that we'll cover during this presentation are pretty high level and apply to all sorts of other automated liquid platforms as well. So for the Hamilton portion of this presentation, I wanted to cover a few different topics. First, talk about the challenges in regards to pipetting, how we account from that from a technology perspective, and then dive a bit deeper into liquid classes before I'll hand it over to my colleague Samantha Turnbull to cover method settings and Labware settings that play a role in facilitating proper liquid handling. Okay, first up, the challenges of pipetting. So suffice it to say, there are some inherent challenges with regards to pipetting, even when pipetting manually. Here you'll see someone's trying to pipette a very viscous solution, glycerin, and you see that it doesn't really get all the way up into the tip very cleanly and doesn't get everything out of the tip. Contrast that with a more volatile, such as acetone, and you'll see that immediately once you're done aspirating, there's pressure buildup that actually pushes the liquid out of the tip in the form of droplets. So there are some liquid properties and as well as environmental conditions that can all impact the liquid handling. And so you have to account for all of these. So how we do that from a technology perspective, most of our pipettes on Hamilton platforms utilize air displacement technology. So if you see the image on the right, you'll see the inside of the pipette has a plunger. That plunger displaces air, and then liquid flows up and collects inside the tip. So this is analogous to that of a handheld pipette, except that on automated platforms, you have the option to take advantage of numerous adjustable parameters to facilitate the liquid handling. Our pipette channels can also accommodate different tip types and tip volumes to handle a variety of applications and different volume ranges. Also, our technologies have a number of different functions for liquid level detection, touch off, side touch, that all ensure proper liquid handling. So let's dive into liquid classes. So we have our eyes and our hands to control and aspirate and dispense action when pipetting manually, but for automated platforms, we have to define parameters in order to tell the system how to pipette correctly. So on our platform, liquid classes are comprised of two things. Liquid parameters that you see here on the right is an example of a liquid class with a list of different liquid parameters, including flow rate, air transport volume, blowout volume, swap speed, and settling time, as well as other parameters. These all play a role in the precision of the liquid handling, how consistent the system is pipetting. The other aspect of liquid classes is the correction curve, which is essentially how we calibrate pipetting channels as it actually adjusts the physical displacement of the plunger for a specified target volume. So we won't have time to cover every single liquid class parameter. So I just wanted to highlight a couple, one that is pretty straightforward and one that is a little less intuitive. So first up is a more straightforward liquid parameter, that of the flow rate and mix flow rate. So the flow rate actually corresponds to plunger speed for aspirating, dispensing, and mixing in microliters per second, and is greatly influenced by tip geometry, viscosity of the liquid, density, and vapor pressure. So pretty predictably, the slower the flow rate would be more applicable to tips with smaller diameters, as well as more viscous solutions, whereas with more volatile liquids, you would want to pipette faster. So have a couple examples here. This showing when you set too high of a flow rate for a very viscous liquid. You'll see that similar to when we tried to manually pipette it earlier, there are some air gaps that are created, and you can see that the liquid wasn't cleanly aspirated inside the tip. And then on the dispense, not everything is ejected out of the tip and into the receiving container. Now, what happens when we really slow things down? So in this case, we have a very slow aspiration flow rate of only 20 microliters per second and a dispense flow rate of 5 microliters per second. You'll see that during the aspirate, slowly the liquid will collect inside the tip. You're not seeing any air gaps or any other disruptions during the pipette. Now on the dispense, the flow rate is very slow so that in this case, the dispense isn't a steady stream of liquid, but is actually just a droplet by droplet. The liquid is being dispensed and emptied out of the well. So slowly but surely, the liquid will work its way out of the pipette. Now, what this mode typically is also characterized here, we're dispensing above the liquid level or into an empty well. There's usually a volume of air called a blowout volume that are also known as like a trailing air gap that helps push the liquid out of the tip and into the receiving container. So one parameter that may be a little less intuitive is that of the stopback volume. This is a liquid volume in which is aspirated immediately after the dispense, and this volume is aspirated as quickly as possible. This parameter is actually only valid and active in jet part mode, which is our mode for multi-dispensing. And this helps prevent droplet formation after each dispense in a multi-dispense process and improves precision overall. So I have a few videos here that show the performance without having any stopback volume set. One example that shows having too high of a stopback volume, and one where the setting is just right. So let's look at an example with a stopback volume set to 0 microliters. You'll see that for each dispense, there's a droplet formation that occurs. So for each successive dispense, the aliquot is having to eject that droplet and move past that droplet, leading to inconsistent performance from transfer to transfer. Now let's contrast that with a higher stopback volume of 15 microliters, and you'll see that while no droplet formation occurs, you'll see that there is a large air gap that's created. Maybe it's kind of hard to see in the video, but you'll see that each of the air gaps at the end of the tip are a little bit inconsistent and uneven from tip to tip. And the successive aliquots are having to transfer past that air gap out of the tip and into the receiving container, again leading to inconsistent performance. Now, what happens when you set the stopback volume just right? In this case, it's 11 microliters. You'll see that there's no droplet formation and there's no air gap. The liquid remains at the end of the pipette tip for each dispense, treating each condition the same and thus ensuring consistent performance from transfer to transfer. So those parameters, along with other liquid class parameters, all play a role in the precision of the liquid handling. In regards to trueness, we have a correction curve. Now, a common misconception when it comes to air displacement pipetting is that the volume of air that's displaced by the plunger is equal to the volume of liquid that you're pipetting. And it's close, but that's not exactly accurate because the volume of the plunger actually has to increment oftentimes more in order to accommodate not only the target volume of interest, but also to accommodate other liquid properties and environmental conditions, so that the plunger actually has to adjust even further to transfer the intended volume. So for manual pipettes, they are actually calibrated, where you have a linear correction across a whole pipetting range. With automated liquid handling on our platforms, a correction curve is used where you can have, for different volumes, you can have a corrected volume for each specified volume. So how does it work? So you would have, for each target volume, would have this corrected volume. So on the right side, you'll see an example of a liquid class, and you'll see a column for target in microliters and a column for corrected in microliters. For the target volumes, you'll see entries for 20, 50, 100 microliters, etc., and then an associated corrected volume for each. So let's look at 20 microliters. You'll see that it has a correction volume of 24.6 microliters. Now, this doesn't mean that when you tell the system to pipette 20 microliters, it'll pipette 24.6 microliters. What this means is that the plunger will increment in order of what it considers 24.6 microliters to be, but physically only 20 microliters is picked up. Now, for any target volumes that actually aren't specified on the correction curve, the software uses linear interpolation between points on the curve. So on the right, you'll see on the graph that for the volumes 20, 100, and 300 microliters, there's a correction for each of those volumes. So if you went to try to pipette 200 microliters, its corrected volume would be interpolated to be 212.5 microliters. Now, through your testing, if you find that when you transfer or specify to transfer 200 microliters, less is actually transferred, say 195 microliters, well, you could add the specific target volume of 200 microliters to your correction curve with the appropriate corrected volume to ensure trueness of that transfer. One other aspect in regards to liquid handling and setting liquid classes is to be aware that our platforms actually have different aspirate and dispense modes. And these modes actually determine which liquid class parameters are active. So it's important to understand how these work, because you could be adjusting some parameters that actually may not even be active during a transfer. Now, for each transfer, you would actually specify a liquid class, and typically for one aspirate and dispense cycle, you would just use one liquid class for that transfer. So we have 4 different dispense modes. The first 2 up here are jet empty and jet part. Jet empty is when you're dispensing the entire contents of the tip above the liquid level or into an empty container. Now, this mode is usually used when you have higher volumes, when you're reusing tips, and is often characterized by a slightly faster flow rate as well as a blowout volume of air to help push that liquid out of the tip and into the receiving container. Jet part is when you're dispensing partial amount of the liquid. You aspirate a total volume and you dispense part of that volume for each transfer. This is the mode that you would use for multi-dispensing. And again, since it's in jet mode, that means it's dispensing above the liquid level or into an empty well. The other 2 dispense modes are surface empty or surface part. Surface empty is when you're dispensing the entire contents of the tip directly into liquid that is held in the receiving container. This is the mode that you would use when optimal performance is required. So this is often the mode that you would use to ensure proper low volume pipetting. It's also the only mode in which mixing is active on the dispense. Then the final mode, surface part, is when you're dispensing a part amount of the total liquid, of the volume of liquid into the well, but dispensing it directly into liquid. So this is used in the rare examples where you might be multi-dispensing, where reuse of the tips is acceptable. Well, now I'll hand it over to my colleague, Samantha Turnbull, to give more insight on method settings and Labware settings.
Thank you, Eric. In addition to all of the properties and parameters that Eric just mentioned, there are a number of settings within the aspirate and dispense commands themselves that will influence your pipetting behaviors. So let's start with the aspirate step. There's a few obvious parameters here. So we have an aspirate volume, aspirate mode, tip type, and liquid class that are defined. But in the bottom half of the dialog, you'll see some other options to describe how to aspirate. So we have a few options that are liquid level detection. This will allow the tips to detect the liquid level during that transfer. So this is best if your source container is a number of sample tubes that have different volumes, or a trough that has varying levels of volume in there. This will compensate for that variability. Once you liquid level detect, you can specify a submerge depth into the liquid before the aspirate will actually start. You also have an option for a fixed height aspirate. So if you have very small volumes or very consistent source volumes, you can use a fixed height. This is a distance off the bottom of your source container in order to start aspirating. Our aspirate step also has a couple important options in this advanced tab. So you can have liquid following turned on. This allows a tip to detect the liquid level with liquid level detection, submerge 2 millimeters, and then stay submerged 2 millimeters throughout the duration of the aspirate command. So this prevents unintentional aspiration of air from the tip leaving the liquid, or accidental over-submersion of the tip, which may cause inconsistencies as well. You also see that there's a pre-rinse mix option here. There's a few liquids that really benefit from a pre-rinse or pre-wet of the tip. Otherwise, you can use this pre-mix to make sure any sample or reagent source that may have settled is fully homogenized before you start your aspirate step. Now in the dispense mode, you will see many similar terms. We have our dispense volume and dispense mode, and we again have a fixed height option, so a distance to dispense off the container bottom. This is best when you're going into empty labware or jet dispensing into the air. You also have the option for liquid level detection. So if you are dispensing into a liquid that is already contained in your target container, you can use that liquid level detection again. There are a couple other modes listed here, touch off and side touch, which I'll go into more detail with in a few slides. Now, all of these aspirate and dispense positions are really dictated by your labware settings. So I can't dispense 1 millimeter off the bottom of a well if I don't know where the bottom of that well is. So especially for your aspirate and dispense positions, in addition to the XY centering of your tip within the well, the Z height for the bottom of your well is very important. So you want to teach the bottom of your well such that the tip is never occluded or blocked by touching the bottom of the well during any of your aspiration or dispense commands. It's best to leave a small gap here to allow free flow of liquid in and out of the tip at all times. Now, once you have a good, accurate labware definition, we also have some other modes you can use. One is a touch off. So this allows our tip to move downward, detect the bottom of the well, and then retract a specified distance before starting the dispense. So this does not excuse a bad labware definition, but it is very helpful when dispensing very small volumes into empty wells, where the dispense distance above the bottom of the well is critical. So if you want to use the interaction with the bottom of the well to wick out that last bit of liquid from the tip, you want to be very, very close to the bottom. Now, this will function very similarly. So if you have a touch off and then a dispense 0.5 millimeters above, it will work very similarly to a fixed height dispense of 0.5 millimeters. However, the physical touch off gives us that confidence that in the moment of dispensing, that well bottom is where we expect it to be. So with very small volumes, the best case I've seen this work is in a PCR plate that's been thermocycled a number of times, where there's a potential for slight warping or there's slight tolerances in the bottom of that well. And so let's look at a video of what this touch off looks like live. Now we're using a much higher dispense volume than you normally would for this touch off, just for demonstration purposes. But you can see that it touched off on the bottom of the well, moves up 5 millimeters, and then starts its dispense. Sometimes you have some low volume pipetting and can't touch the bottom of your well. You already have something contained in there, or for another reason, you can't use touch off. Another option to get that last bit of liquid out of the tip is a side touch. So in this case, we're showing an example in a PCR plate with tapered wells. Again, we're using a much higher dispense volume than we would normally recommend use for with a side touch for demonstration purposes. But you'll see the tip will enter the well and stop a specified distance above the bottom. So in this case, it's 4 millimeters. And then it moves to the right side of the well and touches the wall. And this touch off distance in this case is 2.6 millimeters. Now, based on this, you can hopefully tell that these distances are 100% tied to the geometry of that well. Imagine if your touch off height is slightly different, that touch off distance would need to be different. Or if you had a flat bottom plate with cylindrical wells, it would look very different. And for that reason, these heights and distances are set in the Labware definition itself. This is also where you would enable this side touch off. So this is a particularly helpful technique if you have hanging droplets, if for some reason you can't get that last bit out of the well. So you specify a touch-off height and touch-off distance for your specific container. Now, this is most applicable when the following step is a large volume bulk diluent dispense, or the plate's coming offline to centrifuge or vortex mix, because in that case, in this case, there may be some residual liquid on the side of the well where you dispensed. Now, once you have all these options, they're not always the best case for every situation, but they are great tools in these specific cases. Once you have a good transfer, you're not seeing air bubbles, you're not seeing hanging droplets, everything visually looks consistent, you will want to move on to some verification and quantification of the transfer. So at this point, thank you for listening. I'll hand this off to Nick to go over some more details about some of that verification.
Beautiful. Thank you, Eric and Sam. I really appreciate it. Hamilton is a great partner of ours. I'll be selfish and say I think they're one of the best ALH OEMs that we have as a partner, and have some of the leading technology out there as well. I think from a global perspective, I think Sam and Eric did a great job about getting into the granularity of liquid class optimization. I want to zoom out a little bit and then really talk about volume verification to get to liquid class optimization, and then kind of get into some real world examples, and then really just open it up to questions in general. So with that, let's just talk about liquid handling in general, right? I think we think about liquid handlers, but before you could even get to liquid handler, we have to think about pipettes and people. There's an optimization step on that, because anything that gets to a liquid handler starts off at the bench, right? As a lab scientist for a long time, there's always this war between the automation engineer and the bench scientist. I would transfer it over to the automation engineer, and if it didn't work, I would ultimately blame the automation engineer. But really, I could have been wrong with the transfer steps as a bench scientist as well. So let's kind of talk about it and delve into that a little bit more. So when we did this example at Artel where we took a multinational company and we measured 10 microliters through a whole process from the bench all the way to a liquid handler. Sorry to our Hamilton folks, this wasn't one of the Hamilton liquid handlers. This was a Beckman in this case in general, but we won't say anything. But anyways, we took a look kind of from their manual steps to scale to multi-channel pipettes all the way to the transfer step to an automated liquid handler in this case. We saw variability from site to site, from transfer step to transfer step. So if you zoom out even more, if you're not optimizing at the bench level in the transfer steps and not optimizing from site to site, you'll notice a lot of variability, right? You have some places that you have relative inaccuracies of 5% and CVs that are upwards of 2%. And depending on assay, that could be truly impactful as well. So zooming once again out, we really have to be able to look at the manual steps all the way to the automated steps as well. Now, even thinking about robots, Eric said earlier that most of Hamilton products, and honestly, most liquid handling products in general in the market are air displacement, right? So air displacement is coupled to environmental factors. And positive displacement a little bit different. There's other modes, but I think most of the main commercial liquid handlers on the market are air displacement. So if you take a liquid handler from site to site and try to apply the same optimization settings that you've had for one site, it might not be the exact same for site B or site C. We actually took that example here with one of our forensics labs that was, once again, all across the US. We compared, they had 3 separate liquid handlers across the sites. They've optimized it in city A, and they tried to use the same optimization settings in city B and city C. And they noticed that it wasn't performing to the same specification for relative inaccuracy and precision. So really zooming out and making sure that we're also optimizing from site to site because of environmental factors with air displacement pipetting is super important as well when we look at liquid class optimization. So really high level, that's kind of another way to look at it as well. But once again, in even zooming back more, this is lost as well, but even from a manual pipetting perspective, because most manual processes will be, especially with scale, will be transverted over to some type of automated process. really making sure texts as well are optimized. I joke, there's liquid class optimization and there's people optimization also, right? So if you don't have people optimization and they're not calibrated in a certain sense and optimized by pet, then you're not even going to be able to optimize liquid classes once you even get to a liquid handler as well, because you're going to be chasing a target that's always moving as well. So these are really things to think about when we talk about optimization. There's the liquid class optimization, which Eric and Sam went over really, really well into a lot of detail. But then there's the global optimization from site to site, person to person, and then the method transfer process as well. So where do we come in, right? Why are we here talking today? We have Hamilton, which is best in class when it comes to automated liquid handlers. And I think us as a company with Artel, not to confuse you with the Nova branding now, we're another portfolio under Nova now. But I think we are the best in class when it comes to volume verification. And why is that important, right? In order to optimize, you have to have real-time data with the volumes, right? We have to understand and have just a general foundation of what the volumes are looking like and to trust the reads that we're getting from those volumes as well. I think that's where our technology comes into play. So we have what's called our MVS, our multi-channel verification system. I joke with customers, most people call this the Artel in the field. And if we ever come to your site and you know what MVS stands for, I will buy you lunch. That's how well of a brand name we're known for in the field. But essentially what we do, we have a bunch of different formulas, a bunch of different cool ways to derive our volumes, but we are unlike any, I think, volume verification technology that's out there. We rely on dual dye ratiometric photometry. I won't get in the weeds there. If you want, after this webinar, if there, is more interest in that, we could definitely get into how we do it. But essentially, how we get there is this dual dye ratiometric photometry, and because of that, we're able to go really, really low and also to a relatively good amount. So we can measure anywhere from 10 nanoliters all the way up to 350 microliters really, really quickly. And why is that important, right? Because when it comes to optimization, we really need to be able to optimize volumes with foundational volume settings. So real quick, we're gonna have a polling question. So I will let Ryan put that up here.
So this should be up on everyone's screen right now. And as with the previous one, you can participate if you're watching via GoToWebinar by selecting on any of the answers you see in front of you and clicking submit. The question that we're asking this time is, do you currently have an Artel system in your lab? Your answer options are yes, we use the MVS system. Yes, we use the PCS system. Yes, we use both MVS and PCS. Or also no, but we're considering it, or no, not at this time. So as I said, the previous question, please select one and submit an answer. We'd love to hear from you right now at this time before we continue to hear from our speakers. The question of, do you currently have an Artel system in your lab? Looks like most of you have your answer in. So thank you very much for participating. Let's look at the results for this question. It looks like we have 56% saying no, not at this time, but then 34% for yes, using the MVS system, 6% no, but you're considering it, and then 3% yes for the PCS system. So again, thank you very much, and I'll now hand it back to our speakers.
Thank you, Ryan. Cool. So it's great to see that some of you have the MVS. If you don't, love to have those conversations as well. I will also, credit my Hamilton colleagues as well. There's other ways of doing volume verification with LVK, which is their liquid verification kit as well, which is gravimetrically. And I think both serve really great utilities for most labs as well, especially if you have one of the Hamilton flavors of automated liquid handling systems also. So we really talked that we kind of went back a little bit of optimizing people, pipettes, and liquid handlers. Now I kind of want to walk you through optimizing liquid handlers in general. And I think Sam and Eric went in way more detail than I'm going to go in, so I'm not going to bore you guys here. I will do just a general overview of what we see the most, and then we'll get into a few real-world examples as well. So I always like to break it out into groups here and a couple separate hats. There's verification and calibration of liquid handler, and then there's liquid class optimization. So before we could even think about liquid class optimization, we have to make sure our liquid handler is performing up to the specifications that we think it is. So what do I mean by that? So let's say we have a Hamilton Star. I see a lot of people had one of the Star brand Hamiltons from the polling question earlier. If we dial it into 5 microliters and we expected an accuracy of 5% and a precision of 5%, if we were off at that step, right, just with the general calibration of that liquid handler and volume verification, there's no point in getting the liquid class optimization, because then you are chasing a moving target. So I always like to break these into 2 hats, right? Make sure our liquid handler is working to our specifications first, and then delve into the liquid classes that Eric and Sam were going over in such great detail earlier as well. And how do you do that, right? It takes consistent dispensing, measuring, so seeing what that is, assessing, and then adjusting. And that's where Eric and Samantha really went over, is like, how do we adjust the liquid classes to be within the tolerances that we did as well. So always break it up into 2 hats. I think a lot of customers will get to the liquid class optimization without being able to think about verification and calibration first. And then once again, I think Sam and Eric did a really good job. They went over a lot more steps than I did here. There's a lot of different liquid class parameters that we can adjust to see differential results, right? There's aspiration rate, there's dispense rate, there's trailing air gaps, there's leading air gaps, there's a bunch of other things that I don't include in this, but with each separate parameter, you can adjust and then see how it affects the volume. We kind of did that with this example in general. We had a few different variables that we were looking at, and we adjusted one at a time. And with that, we could see what happened to the liquid class based on those individual variables. So we basically took each of those variables, we optimized it over time, and then we're right around with our last variable in those consistent trends to that 10 microliter mark that we were after in general. But this is big, right? Having an ability to measure those volumes, assess, and then make those adjustments so that we're getting close to the accuracy and the precision that we need to be able to do in general. So this is kind of walking over being able to optimize variable by variable, step by step, with volume verification involved in it as well. And then kind of why we're all here today. It's super important, and I can't stress this enough, to optimize our liquid classes, because volume, and I think people don't realize this, as a bench scientist, I didn't realize this as well. We get so involved in the chemistry or so involved in the pneumatics of our assays that we don't think about volume. Volume is essential to anything that we do in the laboratory. If we're off with our volume, there's no shot that we're going to get close to anything that we're doing. So I just wanted to walk through a few examples just to show you why it's super important to really go over liquid class optimization in your labs and to really incorporate that as part of your processes. So, and it comes to compound management. Those of you on this call that work in this, there's compound volume, there's working volume of what you're putting your compound in for your library. If we think about a couple separate steps, right? Let's say we add 6.8 microliters of our 2 millimolar compound versus 9.97 microliters of our 2 millimolar compound. That can result in a difference of around, let's just round up to about 40% in compound concentration. That's super big. These compounds, are just to do one of these assays can mean multiple hundreds of thousands of dollars. If you're overly concentrating your compound or diluting your compound out, that's super big when it comes to screening, right? Not to even mention the working volume that you're actually putting your compound in. What if you're off by, in this case, we're looking at 40 microliters, but let's say we have a variance of around 4 microliters, plus or minus 4 microliters, right? That could even result in another 50, let's just round to 57% with our compound concentration. So overall, as a whole, right, if we're not transferring compounds correctly, that could result in a greater than 50% concentration impact. And you're going to be chasing what's happening with your compound yields, right? Why do I have a lot of compound or a little compound? And that successful troubleshooting is going to take a lot of impact and a lot of effort to your organization. So this is one of the studies that we did a long time ago. We even have our old transistor logo here as well. So I think this study was done around 25 years ago to show kind of the effects of compound management and if you're off with the volumes of your compound in general. The other thing, and especially with our Hamilton customers who think Hamilton's really, really known for the ability to scale genomics workflows. a lot of the steps in a genomics workflow, so this is an outline of library prep all the way to sequence. There's a lot of processes in this workflow, especially in red, that if you get wrong, you know, you're going to sequence it, you're going to look at the library yields, and you're going to see, you know, sequencing by somewhere, and that's going to cost a lab thousands of dollars of you know, reagents and time, not to mention troubleshooting and going back. All these steps are really important to kind of assess and make sure that you're optimized with those addition of the master mix, the DNA normalization steps, the bead capture, to really make sure that each of those volumes are working sequentially and working accurately and precisely. So once again, I just took an example of one of the labs that we did with a genomics workflow. And this, I think Sam and Eric outlined really well earlier, but a lot of, there's the individual, you know, parameters that you can adjust. And then there's kind of the global parameters with slope and offset. They're different language, they're called different things depending on whatever liquid handler you have. But this is just an example of one of the genomics customers that we had that had, forget what liquid handler, but they were trying to dial in a 2 microliter dispense as well. We basically use the MVS to adjust the slope and the offset to lower their precision and the relative inaccuracy. So before we used the MVS, the relative inaccuracy was around 13%. After the MVS and making an adjustment to the slope and offset, we were able to get it around 2%, which is super big because that 13% was causing a lot of false positives within their assays as well. So just highlighting again another example of what Eric and Samantha covered a little bit earlier as well. And then once again, accuracy and precision super important. There's liquid handlers, you know, not Hamilton, but maybe other liquid handlers that I won't name. They'll even drift on the day-to-day basis or a month-to-month basis. So staying out in front of that is very important. And we've seen this time and time again. You could kind of see inaccuracy drift all over the place with this one example of 50 microliters. Utilizing the MVS, this specific customer was able to track that and optimize almost on a daily basis to make sure the liquid classes were within tolerances as well. All right, now getting back to a couple more examples, qPCR genomics workflow here. So in this specific example, we were taking a look at the setup with a cyber green assay. The control for us was around 1 microliter, but if you were off by 20% with your volume, it would result in 20% inaccuracy. So if you're 0.2 high or 0.2 low, which doesn't seem like a lot at 1 microliter, 1.2 versus 0.8, it had drastic effects on the inaccuracy of volume and drastic effects on the actual qPCR assay in general. So once again, I'm hammering the point here as to why you need to optimize these liquid classes and to always continually reassess them and make sure that that liquid handler is, you know, delivering to what you think it's delivering to as well. Same thing here, you know, there's part of liquid class optimization also involves, you know, optimizing the separate assay parameters. There's mixing and centrifugation. You can see, especially when it comes to qPCR assays or a bunch of genomics assays, there's centrifugation and mixing. We were able to assess that, and you can see with no mix and centrifuge or no mix and no centrifuge that you have the drift of accuracy and imprecision as well. And then once again, the last thing I'm going to kind of talk about as far as optimization goes too, you know, I know Eric and Samantha will say the same thing here, and something for every one of you on this call to think about: using manufacturer tips. So if you're using the Hamilton brand product, using Hamilton's tips versus the third party from overseas, you're really, we see this time and time again going out into the field. Companies are trying to cut costs. Ultimately, they think they're saving on tips, but actually they're creating a lot more inaccuracy and imprecision. So we did this comparison with an ALH vendor tips versus a couple of separate bulk vendor tips, and saw really the precision kind of drift all over the place with the bulk tips versus the ALH vendors as well. Now, that's not direct optimization with the liquid classes, but that's optimization of your workflow with a liquid handler in general. Best is vendor tips. We have a bunch of data to prove it, and we have a technology to prove it as well for you to assess the tip types also. So why is this important to all of us here? Why are we here today? One, Hamilton did a great job at saying, how to optimize liquid classes. I kind of walk through, globally why you do it as well. But ultimately, it comes down to costs, right? Liquid handlers are not cheap to run. The reagents dependent on assay types are not cheap to run as well. So making sure we're optimizing our liquid handlers are super important because the cost burden on different assays can become astronomical. If you think about most genomics workflows or even sequencing workflows, if you're off with your liquid handling and you find that you're off and you haven't optimized, that could be super substantial to your org and cost you guys a lot of money in troubleshooting and going back and seeing what happened and then reworking everything. So it's really important early on to establish these processes, whether it be with Hamilton, with us, or with both of us at the same time, because we're really here to help you. Once again, I said this earlier, and I'm not saying this just to be nice because my Hamilton colleagues on the phone, they have some of the best in class liquid handlers in the world. And I think we have some of the best in class volume metric verification tools in the world as well. So working together and establishing this foundation early on will really help you save a lot of time and money as you scale your processes as well. And lastly, right, I think it all gets back down to it. This was all cool, what we did today. We talked about this, but really, I want you guys to always think about what happens if your assay fails. Go back to your volume verification processes, right? How do you ensure that your liquid handlers are consistent across sites or labs? That's why Eric's team is here. That's why he has over 80 apps people in the field. They're really here to help you, and we're really here to help you as well, establishing liquid handler types on all your different locations as well. And lastly, and I think this is a big thing, I don't know if many people on this call know, but ISO 23783 got released in 2022. That is basically compliance around automated liquid handlers in general. Normally, it takes a few years for compliance to scale. So we're in 2025 right now. It's not fully being enforced, but I do know as we start to get further away from 2022, maybe next year, maybe 5 years from now, that will become a bigger part of being in compliance. So we're here to help you with that as well. From Hamilton to Artel, being in compliance is something that we always think about and stress as well. So with that, that was a whole lot of information that Eric, Samantha, and I covered. I think we went over, globally, how to optimize liquid classes. Thank you for the granularity from Eric and Samantha. And then I really wanted to cover the why it's super important and how to do it also. So with that being said, I'll turn over to Ryan. Questions, concerns, anything that Eric, Samantha, and I can answer.
Yes, well, thank you so much, all of you, for that very insightful presentation. As we move into the Q&A session, I do want to direct the audience's attention towards the handouts module, where you'll find some additional documentation related to today's presentation to download for some further reading. I'd also like to invite the audience to keep sending their questions or comments right now for this Q&A portion of the webinar. If you're attending via GoToWebinar, please use the questions window, and those watching on LinkedIn Live events, feel free to post your questions in the comments section. Now, I've already received some questions, though, from the audience, so we'll get ourselves started with those. The very first question I have here, it says, it just wants to know, what are the most impactful liquid class parameters that one should consider?
I can take that one, but my answer is going to start with it depends. It depends on exactly what you're doing. So if you're seeing hanging droplets, well, you're probably going to need an air transport volume or a leading air gap to help accommodate that. If you have too much residual liquid that's being left inside the tip, you're probably going to have to implement a blowout volume of air. So it really is dependent on the context in which you're utilizing, these steps to focus on which parameter of interest. Definitely, flow rate, as we saw demonstrated in the videos today, is an absolutely critical parameter. But for our platforms, know that when you install the software, it comes with a liquid class database that has liquid classes predefined for all different sorts of liquid types. So you already have something to start with, and along with the proper method settings and Labware settings that Samantha covered, you're going to be in the ballpark of achieving a proper liquid transfer.
And I echo that from Eric as well. You know, and I think Hamilton does a really good job at this with the pre-established liquid classes already in there. But it gets you close to what you're doing, and then it's up to you to really optimize it. And it's really different, like we covered earlier. It's really dependent on lab, assay, even, you know, Samantha covered a little bit earlier, tip touch. There's certain times that you can't tip touch depending on your assay though too. So it's really, you know, lab dependent and assay specific as well.
Excellent. Thank you so much. The next question then here is wondering, where can I learn some more about optimizing automated liquid handling?
Ooh. I think we both have different ways that both of our companies do that as well. I don't know, Eric, if you want to start, and then I'll come off your pigtails.
Or Samantha, you want to chime in?
Sure. Hamilton has a lot of great resources on our website. We have some training resources. There's definitely reference that handout that we've attached, which is some liquid handling references, really describing those modes and parameters. But it's really about understanding your platform, what automated liquid handler you're using, and what is important to that platform. So definitely recommend some training. We have some other webinars we've gone over specifically about liquid handling that are available on demand on our website, and some specific for applications. So if you're looking at NGS or you're looking for specific applications, we have some guidance there as well. What do you got, Nick?
Yeah, same here. We have a bunch of different resources, a bunch of different case studies, and we even have Excel sheets that basically, you know, for the main flavors of liquid handlers, from Hamilton to Beckman to TCAN, if you throw in your slope and offsets, we'll tell you exactly how to adjust them as well. Also, shameless plug here at SLAS this year with our partners at Hamilton. Every year we do a liquid handling essentials course, and we have this year is a little bit different for us as well. We have the morning session will cover just general liquid handling essentials. So it's actually a workshop. We go through a bunch of didactic presentations, but then you get hands-on time with the liquid handler. So Eric's been there for, God, how long, Eric? You've been there over the last decade helping us with that. And so you'll get hands-on with Hamilton, Agilent. We have a couple other vendors, and then even in the afternoon we have a more in-depth course where this is the first year we've done it, where we get really into the nuances of what Samantha and Eric were talking about today as well. So we have a whole bunch of collateral that we could share with you from both companies. And once again, shameless plug, we even have one of our workshops at SLAS coming up in, I believe it's end of January, but I could be off with that.
That's great. Thank you so much, all of you, for your thoughts on those questions. But we have reached the end of our time here today. Now, if we couldn't get to your questions, know that the team here will follow up with you. Or if you have some further questions, you can direct them to the email addresses that are up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and then gain access to features such as viewing the recording and more. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks, Ryan.